Hi everyone, hopefully you can hear me in a moment, and what we're going to do is we are going to go through all of the content that could come up on the B1-3 paper tomorrow. So the way this is going to work is I'm just going to go through the whole lot. We're going to start with B1, work our way through B2, B3, and I will try to flag up any of the bits that are on the advanced information, but because it does have that lovely little statement on there that says there is no topic that won't be examined, or whatever they'd like to say, then we'll cover everything, because there's probably going to be a mark on everything somehow, so we'll do the whole lot so you're well prepared. If you do have questions, you can ping them in the chat window and I will try to answer as many of those as I can. So let's make a start then. Here we go. B1 it is then. So the first thing we actually looked at in B1 were the different types of cell. And hopefully we remember that as far as cells goes, we've got two types. We have prokaryotic cells, which are the bacterial ones, and eukaryotic cells, which are going to be the plant and animal cells. We need to be able to compare our prokaryotic and eukaryotic cells. So as far as prokaryotic go, they do not have a nucleus. They are simple cells, approximately 1 micrometer to 10 micrometers in size. Eukaryotic cells, however, they do contain a nucleus with the genetic material in there. They're more complex and they're larger, 10 micrometers to 100 micrometers. And just remember that little maths link that 1 millimeter is the same as 1000 micrometers. Which brings us nicely on to our first cell. So what we have is our animal cell. Now, the animal cell has 4 important parts that we need to know, okay? So the 4 bits on the animal cell are the nucleus, the cell membrane, the cytoplasm, and then the mitochondria, the little dots in there. So what we actually have there then, just 4 parts that we need to know. Please don't ever write that there is a cell wall in an animal cell. It is only a cell membrane around the outside. Our next cell is the plant cell. So our plant cells then have a little bit more on there. We have got around the very outside the cell wall. The next one in is the cell membrane. We still have our cytoplasm here, the kind of yellowish thing on this little picture. Nucleus. The green splodges are our chloroplasts, and we have our vacuole. The little dots, again, the mitochondria there. So we do have some similarities between our plant cells and our animal cells. They both have a nucleus, a cell membrane, the mitochondria, and a cytoplasm. But our plant cells also have the cell wall, the chloroplasts, and the vacuole. We need to know what they do. Okay, so as far as the nucleus goes, that's all to do with controlling the cell activities. So if you're asked what does the nucleus do, control cell activities is absolutely fine. Cytoplasm is where the chemical reactions take place. The cell membrane is what's going to control what enters and leaves the cells. Your mitochondria is where aerobic respiration takes place. Cell wall is all to do with making the cell rigid. So it's all about supporting the cell, and it's made of that stuff called cellulose in our plants. Chloroplasts contain chlorophyll, which traps the light energy for photosynthesis. And the vacuole, that contains cell sap, which is all to do with expanding out against the cell wall to help support it and allow it to grow nice and vertically without a skeleton. Lignin is going to come later when we actually look at the xylem, so bear with us on lignin. Prokaryotic cells then. Here's a lovely little diagram of a typical prokaryotic cell. Now, what we actually find here is there are certain features in common. We still have cytoplasm, we still have a cell membrane, and some of them also have this cell wall here as well. But then we have new features that are only in our prokaryotic cells. So first of all, no nucleus. The genetic material is just in a loop here, just free floating in the cytoplasm. We've got these additional little loops of DNA called plasmids. And then we also have these things off the back, the flagella, that's to do with allowing bacteria to move. Some of them will also have this thing called a slime capsule, which just helps prevent them drying out. And the pili, which are all to do with attaching to surfaces. Now, as we've said, bacteria are much smaller. And as we're going to come on to in the Unit 6 work, we look at the fact that we grow them on this stuff called an agar plate. Now, we've already covered most of the actual functions of those subcellular structures in the bacterial cells. Quick recap. Our genetic material obviously contains the DNA that's going to be useful for those genes that produce the proteins they need. Cell membrane controls what substances can enter and leave the cell. Cytoplasm, where the chemical reactions take place. Cell wall is all to do with still holding the cell together and protecting it, but in our bacterial cells made of a stuff called peptidoglycan. Flagella allows the cells to move. Pili allow it to attach to surfaces. Slime capsule prevents it drying out and helps it stick to smooth surfaces. And the plasmid is extra DNA basically. Which brings us very nicely onto our first PAG. So we know there are going to be certain practical tasks they ask us about. And one of the ones listed for both foundation and higher are microscopes. They're not going to ask you about the electron microscope as far as the PAG goes, because we mostly probably go to state schools. We can't afford such things. We have the lovely light microscope, this thing here. Obviously, you will have seen a version of this at school. Depends on obviously which types you have. If you've got the slightly fancier ones, they've got a little LED light at the bottom. If your school is still using some rather older ones, then this will probably be a mirror attached by a bit of blue tack. But our key pieces that we need to know. Up the top, we have our eyepiece lens, the bit we look through. We then have this little dial here, which has the 3 or 4 different objective lenses, and we can just twist those round to select different ones. We've got the 2 little dials on the side, coarse focus and fine focus, and we've got this bit here, which is the stage where the slide sits. So those are the key bits of our microscope that we need to know. Now, we're going to use the light microscope in order to be able to see things that are too small to see with the naked eye. As we know, 10% of the biology paper could be maths-based questions. So one of the maths questions they could ask us here is something to do with magnification. So to work out the total magnification, all we need to do is the eyepiece lens magnification times the objective lens magnification. So simple multiplication. If they were to give you one of the lenses and the total magnification and ask you to work out the other, total divided by whatever lens they gave you. Okay, so don't be thrown by that, and it should be nice and easy. Remember, you're allowed a calculator in every science paper. So how do we use our microscope then? Put your slide onto the stage first of all, and we're going to then select the objective lens with the lowest magnification. So you always start with the lowest magnification. Make sure your stage is up as close to the actual objective lens as you can. Then look through the eyepiece, and then you turn your coarse focus dial until the object becomes clear. If you need to tweak it ever so slightly, use the fine focus, and then all you need to do is repeat that, just twisting to the next objective lens each time. Nice and simple to do. Another maths question they could ask us here is all to do with working out the image or the actual size. So what we actually find then is to work out the magnification, if they've given you the image and the actual size, is just image divided by actual. If they want you to work out the actual size, then what you do is the image size divided by magnification. So if you remember I am, then I, image size at the top of your triangle, A, actual size, and M, magnification at the bottom. You can rearrange no matter what they give you. Because this is a PAG, it wouldn't surprise me if they're going to put in something about stains. Because what we actually find is we've got to stain our specimens because most cells are colourless. So we use a stain in order to make them easier to observe because they provide contrast. Now, what we can do with these stains is we can either colour the whole cell or we can highlight specific subcellular structures. So there are 3 stains that you should be aware of. Methylene blue, that's the one we use in animal cells to show up the nucleus. Iodine shows up the nucleus in our plant cells. And then crystal violet is the one that we use to stain bacterial cell walls. In order to apply the stain, quite simply, specimen on a slide, add 1 drop of the stain, and then we paste a cover slip on top. Now that word cover slip is important. It's been a 1-mark question in the past. So that's the little bit of glass that sits on top of the slide, on top of the specimen there, cover slip. You can't call it a little bit of glass, square of glass. anything else you might imagine, Coverslip is the only accepted answer there. So that covers anything I can think of that they could put onto our microscopes pack. What we're then going to have is the potential follow-up questions to do with the electron microscope. Now, the key thing here is this phrase resolution. When we talk about resolution, we're talking about the smallest distance between 2 points where we can still see them as separate entities. As in, if we've got two little dots, how far can they actually be from each other before they merge into one? Okay. Now, what we find is a light microscope, if they're any closer than 0.2 micrometers, then it's not going to be visible for us. So we need to then trade over to the electron microscope. Now, since we got the electron microscope, we can see things in a lot more detail. We've got more information about what those subcellular structures in our cells are, and we've understood things like viruses, which are too small to see under a light microscope. As far as the electron microscope goes, we have two types. Transmission electron microscope, or the TEM, and this is the one that produces the most magnified images. We then have the scanning electron microscope, or SEM, and this is the one that can produce three-dimensional images of the surface. So what we can do is have a comparison question between the light and the electron microscope here. So what I'd always suggest you do is remember kind of three things that we can then put as the kind of comparison points. So light microscopes are cheap and they're going to be small and portable. So there's your first two. Cheap, small and portable. Third one, I'd suggest is the resolution of our light microscope is only 0.2 micrometers, but we can view specimens that are living or dead, okay? Whereas the electron microscope, it's the opposite. They're expensive, they're large, you can't take it out in the field for sure, but they have a better resolution because it goes down to 0.1 nanometers. However, the specimens are dead. So just go with a couple of those to remember. As I say, 3 is always a good number because if they give you then 1 in the question, you still have 2 to back yourselves up with. Now, next up we are coming on to is the DNA section. So this is listed as both in the foundation and the higher advanced material. So we know something in the DNA bit is coming up. What it is, who knows? So first thing we need to know then, DNA is found in the nucleus. That DNA is then organised into these structures called chromosomes, and those chromosomes are divided up into individual little units called genes. And those genes code for a protein. So what we actually find here then is they have had a question in the past where they asked you to put the different sort of genetic terms in order of size. Genome, always the biggest, that's all of the genetic material in a cell. Chromosome is next, then we come to our genes, okay? So just make sure that you know how it breaks down. Hopefully we know that absolutely everyone has unique DNA unless they happen to be an identical twin. If you're an identical twin, sorry, you're not unique, you share your DNA with one other person. Now, when we talk about our genes, these are where we get all of our kind of individual characteristics. And we're going to be looking at that in more detail in the second biology paper in the genetics topic. But each of those genes is a small piece of DNA that codes for a protein. And those proteins are what actually give us those characteristics. So what we find is that even though every cell has a complete set of your DNA, it's not all expressed. So not all of the genes are switched on. So this comes up as a higher tier question every once in a while, asking why not all proteins are expressed in all cells. And the simple answer is that some genes are switched on, other genes are switched off. Hopefully we do remember that DNA is a polymer made up of 2 strands in that special shape called a double helix. The individual monomers that makes up that polymer are nucleotides. So the monomers of DNA are nucleotides. Each of those nucleotides is made up of 3 parts, the sugar, the phosphate, and then the base. We have 4 bases, A, T, C, and G. You don't need to know the full names, just A, T, C, and G are absolutely fine. Now, what happens is we then end up with these 2 strands joined together in that double helix, by complementary base pairing. So if they ask you to describe the structure of DNA, then double helix is the first thing, and then what we're going to talk about is the fact that we've got complementary base pairs between them, A and T, C and G. That usually covers all three potential marking points that they're going to give you there. If you're doing foundation, this next bit is not for you. This is for the higher tier folks only, and it's how we make proteins. So protein synthesis is a two-stage process made up of transcription and translation. So step one is going to be our transcription. Now transcription is where we are going to make something called mRNA, messenger RNA. So what happens is we've got our entire length of DNA and we unzip the DNA in the region of the one gene we want to make the protein for. So what we find is the DNA unzips or unwinds, either accepted, and then we have free RNA bases that come in and form complementary base pairs, just as we've seen before. So C's with G's, T's and A's, but we will not have any thymine on mRNA. Anywhere that you would have had a thymine, the T, it's got to be a U, because there is no thymine in RNA. Okay, so just watch out for that, because they could very well give you a little section of DNA and ask you to write the complementary mRNA sequence. So in that case, all you'd have to do, obviously just match your C's and your G's together. Anywhere that there's a T on the DNA, put an A on the mRNA, but anywhere there's an A on DNA, put a U on the mRNA. Once we've actually gone all the way along our gene, that detaches and the mRNA then leaves the nucleus, and we then come on to step two, which is translation. So translation happens at the ribosomes in the cytoplasm. So here's our strand of mRNA, and what happens is it's going to be read in sequences of 3 bases called a triplet codon. So we've got our little triplet codons here, and then in the cytoplasm we've got these little structures here called tRNA, transfer RNA. They have an anticodon, which is complementary to the codon here. And onto the tRNA is an amino acid. So every amino acid has a particular little triplet sequence, and that's going to correspond to the complementary one here. So the ribosome comes along, reads the first triplet, the relevant tRNA comes in, and there's our first amino acid. The ribosome slides along, next amino acid is brought in by the tRNA, 2 amino acids are joined together, and it just keeps going all the way along until we've replicated the entire protein chain. At that point, it's released and the protein folds up. So that is all we're talking about with protein synthesis. First step is going to be our transcription, making of the mRNA. Second step, translation, how we actually convert the mRNA into the protein itself. Our next little section is all about enzymes then. As far as enzymes go, these are proteins, first of all, and that means they are susceptible to things like temperature changes, pH changes, etc. Now, because they're proteins, that means they're made of a sequence of amino acids, and they've got a very specific shape. The key thing here in terms of their shape is what we refer to as the active site. So when we look at our enzymes, then there is one very specific shape part of the molecule, which is the important bit. So when we're actually looking at it, we have the active site, which has a specific shape, a shape that's complementary to the substrate. Now, what that means is each enzyme is only going to be able to bind a specific substrate. So what happens? When the enzyme and substrate collide, they form an enzyme substrate complex. They then either build up or break down those particular bits, and then they release them. At that point, the enzyme's free to catalyze another reaction. Which brings us nicely onto a term we can use to describe enzymes, which is they are biological catalysts. So that just means that they're going to speed up reactions in living things. We need to know about 4 of the different factors that can affect the rate of an enzyme reaction. First one, temperature. With our temperature, you would expect a graph like this. Now, you need to be able to explain what's happening here. First bit, as we increase the temperature, we increase the rate of reaction. Reason for that, the particles are gaining kinetic energy, there are more frequent collisions, therefore they will react faster. Top of the graph, this little peak here, that is the optimum temperature. That's where the rate is the fastest. As we go beyond the optimum and keep heating it up, we are going to then denature the enzyme. And by denature, what we mean is we're actually going to change the shape of the active site. So if we change the shape of the active site, the substrate can no longer bind, therefore it just doesn't work. The next one is pH. So here's our pH graph. It's typically a bit of a bell-shaped one because what we find is we have an optimum pH and that pH will depend on where the enzyme is designed to work. In the stomach, it'll be around about pH 2. In other regions of the body, pH 7. Some might be even slightly alkaline. But it does depend on where in the body is as to what the optimum pH is. If we then either go too acidic or too alkaline, we denature the enzyme. So active site changes shape and the substrate no longer fits. The last ones are concentrations. So what we're going to see here, we've got enzyme concentration and substrate concentration. Now, both of them, when you first increase, then we see an increase in the rate of reaction, because if we increase the concentration of the enzyme, then quite clearly there's going to be a much faster rate of reaction, because there'll be more frequent collisions. But we get to a point where actually all the substrate is used up incredibly quickly, because we've got so many enzymes, literally, as soon as they come into contact, there we go, job done. In terms of our substrate concentration, it increases initially, because as we're increasing the substrate, then obviously there's going to be an increase in the rate of reaction because there's more likely to be collisions. Then we get this plateau, this leveling off. That's because at that point, all of the active sites are already in use. So increasing the concentration of the substrate further is going to do nothing. Now, enzymes are another one of those practicals that could come up for the higher tier folks. Realistically speaking, then the enzyme practicals could be graph based. They could be obviously asking us in terms of water baths could come in if we're talking temperature here. So in terms of our water baths, remember we have two key options. The first one is we can heat stuff up using a Bunsen burner. Terrible, rubbish, inconsistent. So if we're asked to suggest problems with that, then first of all, there's the risk of burns. Secondly, you're really going to struggle to keep it at a consistent temperature. So the improvement, we use an electric water bath because we just literally set it to whatever temperature we want and there it will stay. So that covers all of our little bits on enzymes. Okay. Next up, we're on to respiration. So respiration is all to do with providing the energy we need in our body to carry out all of the different reactions. Okay. Now, we need to remember that not everyone needs the same amount of energy because different people will have different energy requirements based on their age, their lifestyle, their gender, etc. So depending on how much activity you put in, obviously determines how much food you then need to eat. We then need to know about some key food groups, and this comes into our biological molecules testing, which is a PAG on both of our papers. So first one, carbohydrates. Carbohydrates are polymers made up of simple sugars. They're mainly used for energy and we will break them down into those simple sugars using a carbohydrase enzyme. Now, what we actually find is that there are two different biological molecule tests associated with carbohydrates. First is using iodine to test for starch. So the iodine goes from orange-brown to blue-black. Or we could test for the sugars themselves using Benedict's reagent, which starts off blue and then it will go orange, red, a different colour. Okay, so those are our first two tests there. Second biological molecule we need to know about, the proteins. Proteins are again polymers. They are made of amino acid monomers and they are broken down by protease enzymes. In terms of testing for proteins, we use something called Biuret solution. Starts off blue, goes purple if proteins are present. Last one is the lipids. Now, lipids are not polymers. They are the only one that are not polymers here. Still broken down by an enzyme, but this time they're lipase enzymes. And our lipids are made from 1 glycerol and 3 fatty acids. So you've got your glycerol and then 3 fatty acid chains joined onto it. When we break them down, what we're going to see is a decrease in pH, because as we break down a lipid, what we're actually doing is releasing fatty acids. And as the name suggests, they are acids, so the pH decreases. To test for lipids, all we actually need to do is add some ethanol to it, give it a little bit of a shake, and if you form an emulsion, you have a lipid. If you don't have a lipid. We need to know about 2 types of respiration, aerobic and anaerobic. So aerobic respiration is the one that's going to be taking place inside all of your cells. You are currently carrying out aerobic respiration. Now, aerobic needs oxygen. So in order to carry out aerobic respiration, we need plenty of oxygen there. It's going to take place inside the mitochondria, as we said, and this is going to then show where we've got cells that are producing lots of energy. If they've got lots of mitochondria, we are going to be producing a lot of our energy. So muscle cells, liver cells, loads of mitochondria, because we have lots of energy required there. As far as aerobic respiration goes, you should know both the word and the balanced symbol equation. Word equation, glucose plus oxygen makes carbon dioxide and water, okay? Hopefully we know glucose, C6H12O6, plus oxygen, O2, makes carbon dioxide, CO2, and H2O, water. To balance it, dead easy, chuck a 6 in front of everything except the glucose. This is an exothermic reaction, and it's worth knowing that aerobic respiration produces a lot more ATP than anaerobic. Anaerobic respiration is our second type, and this one is going to occur where we don't have much oxygen available. So instead of being able to oxidize the whole thing, what we're going to do is we're going to change our glucose to a substance called lactic acid. Now, lactic acid is not great to have build up inside your body because it does cause muscle fatigue and cramps. And what we end up with is something called an oxygen debt. So oxygen debt just means that after you've finished whatever activity it was that created the lactic acid, you've got to break it down. And to do so, we need oxygen. So after exercise, you'll keep kind of breathing rapidly and your heart will beat faster for a bit because it's going to be getting that oxygen in to break down that lactic acid we've created. We still want anaerobic respiration though, because it does give us that little bit of energy when we don't have enough oxygen available. Rather than trying to run from some kind of a threat, you run out of oxygen, then that would be it, okay? This gives you that little bit of extra time to get away, in theory. Second example of anaerobic respiration is fermentation. So glucose is then changed into ethanol and carbon dioxide. ATP is literally the energy currency of the cell, if you like. So ATP is a biological molecule that allows you to transfer energy from one place to another. Next up is photosynthesis, so the last bit of B1. So photosynthesis is all to do with our plants. What they're going to be doing is taking in 2 important things: carbon dioxide from the air, entering through the stomata, and water from the soil, entering the roots by osmosis. From that, we're obviously going to join them together to make glucose and oxygen. Now, this is going to take place inside our chloroplasts, and the chloroplasts contain this green pigment called chlorophyll, which traps the light energy from the sun. This is a 2-stage process. So stage 1, light dependent. This needs the energy from the sun to actually split the water molecules into the oxygen and the hydrogen. Step 2 is going to be the light independent reaction, and that's where the carbon dioxide joins with those hydrogen ions in order to make the glucose. So 2 stages: stage 1, light dependent; stage 2, light independent. It is worth remembering that photosynthesis is an endothermic reaction, so that means it's going to take in energy from the surroundings to do that. In this case, energy from the sunlight, obviously. Once we've made that glucose, we can use it in respiration, we can use it to build cell walls, we can use it to create proteins, it's incredibly versatile. So photosynthesis really is the foundation of a huge amount of things in biology. There are a whole slew of photosynthesis experiments that we need to be aware of. So the first and foremost thing to know here is how we test leaves for starch. And quite simply, take a leaf, chuck it in a beaker of boiling water for a minute just to kill it off. Then what we're going to do is place it into a tube containing ethanol, and that makes the chlorophyll leach out. What we then do, spread it out onto the white tile, give it a rinse, and add a few drops of iodine. Anywhere that starch is present will go blue-black. Anywhere no starch is present stays orange-brown colour. Before we do any investigation into photosynthesis, we need to carry out a process called destarching, which basically means stick a plant in a cupboard for 24 hours without light. Literally all we mean by destarch the plant. If we wanted to investigate that chlorophyll was needed for photosynthesis, then we would destarch a type of plant called a variegated plant. So that's one with both green little white bits on it. So stick it in a cupboard for 24 hours, take it out, put it in the sunlight, test the leaf for starch. You'll find that there's only starch in the green bits. If we want to prove light is needed, destarch the plant, cover a bit of it with a bit of card, leave it in the sunlight for a few hours, remove the card, test the leaf for starch. If you want to prove carbon dioxide is needed, destarch the plant. It's getting repetitive, isn't it? You cover the little bag, the plant with a polythene bag, and you add a little pot of soda lime in there. The soda lime is going to remove the carbon dioxide. So leave it in the sunlight again, test the leaf for starch. If you wanted to prove oxygen is given off, it's different. We don't actually stick the plant in the cupboard for the first time. Instead, we're using pondweed. So to test the oxygen, we need to collect it, and it's far easier to do with something in water. So a bit of pondweed, chuck it in a beaker, stick a funnel over the top with a little boiling tube or test tube filled with water, and then as it photosynthesizes, the gas bubbles go up into the tube, pushes the water out, and you test it for oxygen, which hopefully we remember is a glowing splint will relight. We can investigate photosynthesis as well. Now, this isn't listed as a specific bit on the exam papers as a PAG, so we're going to go through briefly. If we wanted to investigate photosynthesis, then obviously you could just set up an experiment, usually with pondweed, to do this. Now, there are 3 limiting factors that we should be aware of here. First one, light intensity. Second, the concentration of carbon dioxide. And third, the temperature. So these are the factors that could limit the rate of photosynthesis. Now, as far as light intensity goes then, we would have a graph like this. So as we increase the light intensity, the rate of photosynthesis increases. But we hit a point where it levels out, it plateaus. And that's because at this stage, something else is limiting the rate of photosynthesis. If we then come on to carbon dioxide concentration, increasing the concentration of carbon dioxide increases the rate of photosynthesis, but it levels off again. So here, something else is the limiting factor. You're going to sense there's a recurring theme through those 2 graphs, okay? So always increasing the factor increases the rate of photosynthesis, but where you get any horizontal part, that just tells us that there is something else limiting the actual rate. So there is another limiting factor at play. Third one is our temperature. This should look familiar to us from our enzymes. It's exactly the same because photosynthesis is an enzyme controlled reaction. So we increase the temperature, rate of photosynthesis increases initially because they're going to be gaining kinetic energy, more frequent collisions. Hit the optimum, fastest rate. Beyond the optimum, we denature the enzymes. Therefore, rate of photosynthesis drops right off. So hopefully you can explain how they interact with each other. They could, in theory, ask us a kind of practical based question here if they're looking at the kind of best ways to measure gases. So simple way, count the bubbles. Slightly better way, collect the gas in an upturned measuring cylinder. Best way, use a gas syringe. Counting bubbles sucks because the bubbles are different sizes, you're likely to miss them when they're going super quick. Upturned measuring cylinder, good luck getting that completely full without a little air bubble in it. So the gas syringe is by far the best method. Now, obviously for the higher tier folks, the last thing you need to know about is the inverse square law. And quite simply, that just tells us that when you double the distance from a light source, the light intensity decreases by a factor of 4, which we could actually write as a mathematical term of 1 over d squared, where d is the distance from the light source. So just remember that little pattern there in case it comes up. So double the distance, light intensity decreases by a factor of 4. So that is all of B1 done, okay? B2 is next. So B2 kicks off with our 3 key processes that we're going to be able to basically move substances around. So first one is diffusion. Quite simply, diffusion is the movement of particles from an area of high concentration to an area of low concentration. You could phrase that as they move down a concentration gradient. So because they're going down the concentration gradient, this is a passive process. It does not need energy. 3 factors will affect the rate of diffusion. First one, distance. So the smaller the distance, the faster the rate of diffusion. Second, the concentration gradient. Steeper gradients lead to a faster rate. And the third one, surface area. If it's got a larger surface area, then we've got a faster rate of diffusion. Okay, so 3 ways that we can affect the rate of diffusion, and this is going to come into play as we look at some of these specialized surfaces. Process number 2 is osmosis. Please be careful with the definition here because there are 2 parts to it that we need to get right. Firstly, osmosis is only to do with the movement of water, so don't forget that bit. Anything else, not osmosis, only water. So, definition for us to learn, the movement of water molecules from an area of high water potential to an area of low water potential, second part of the definition, across a selectively permeable membrane. Please don't use other words from selectively, so they will not credit other ways of writing that. Selectively permeable membrane is the best answer to give, guaranteed marks. Now, what we actually find is that plant cells and animal cells behave in different ways here. If we're thinking about animal cells first of all, if we put them into our distilled water, say, okay, so distilled water has a high water potential. The cell has a lower water potential. So water's going to move into that animal cell and it's going to swell up, but it will eventually burst in a process called lysis. If we were to do the opposite and place it into quite a concentrated sugar solution, then what's going to happen is it's going to draw water out of the actual animal cell and we end up with it being crenated. So basically it shrivels up. Neither of those is a good scenario to have. Plant cells are actually a little bit different. If we've got water moving into our plant cell, it's not going to burst because of the cell wall. So instead, the vacuole swells up, pushes out against the cell wall, and the cell becomes turgid, okay? If we go the other way, water's being drawn out of that plant cell, then it becomes flaccid first of all, and if it keeps drawing out water, the cell membrane actually gets pulled away from the cell wall and the whole thing collapses in a process called plasmolysis. Third process is active transport. Now, active transport is the different one because this one is going in the opposite direction. So this is where we're moving substances from an area of low concentration to an area of high concentration. So they're moving against the concentration gradient. Because of that, they need ATP. Okay, so it's an active process that needs ATP to actually allow us to move those substances in basically the wrong direction for them. They also require these things called carrier proteins in the cell membrane as well. So because of this, any of those cells that carry out active transport will have more mitochondria present as well. 3 kind of key places that we will see active transport taking place. Small intestine to take up glucose, nerve cells where we're going to be moving potassium and sodium ions, and then in our root hair cells to take up the minerals from the soil. Our next little bit then is the process of mitosis. Now, we only have to do mitosis for paper 1, meiosis is the second one. First and foremost, when you are writing the word mitosis, make sure there is a letter T in it. If the T is not there, they will not give you the mark because they will think you're writing meiosis. So the T is the most important bit of your spelling, if you like. Mitosis is going to be used to produce more normal body cells. So we use it to repair damaged tissue, to replace any worn out cells, to allow organisms to grow. Always comes back to mitosis. And what we're going to produce as the end result are 2 genetically identical daughter cells. That's a key phrase, genetically identical. Don't just write the 2 cells are the same. Let's get into the habit of writing properly, okay? Genetically identical cells. When we come on to this whole process of how we make these cells then, we go through something called the cell cycle. Now, the cell cycle basically has 4 kind of key parts to it. First one is we have a growth phase. Secondly, we have our DNA replication, or the S phase. We then enter our mitosis, or M phase, and we've got growth. So 2 growths, mitosis, and our synthesis phase. What we're going to do is we're going to focus on the DNA replication and the mitosis bit. They're not going to ask you exactly what happens in the growth phases. They just exist around things, if that makes sense. So, to replicate DNA, first thing, the DNA double helix is going to unzip. We form single-stranded templates, and then DNA bases are going to come in and form their complementary base pairs, A and T, C and G. So first mark, DNA unzips. Second mark is for saying complementary base pairs. Third one, you can put down A and T, C and G. That's going to give us 2 complete sets of DNA inside our nucleus here, okay? Then we come on to the actual process of mitosis, which is how we're going to actually split up these chromosomes to make our 2 sets. So what we've got is this. We've obviously started with our normal cell here. We're going to copy all of our DNA. That's what we just did. So the DNA unzips, the bases come in, and they form their complementary base pairs, A's and T's, C's and G's. 2 complete sets of DNA are now present. The next thing that happens is the nucleus is going to break down, and our chromosomes are going to line up along our equator. If you can't remember the word equator, middle of the cell works. So the chromosomes line up along the equator, and then they are pulled to the opposite poles down here. So we can see that they're being pulled to the opposite poles, or opposite ends if you prefer. And that's done by these little things called spindle fibres. What happens is they go all the way to the opposite ends of the cell, and then we end up with the cell dividing in a process called cytokinesis. Now, cytokinesis quite simply is where we've got our cell, and then the membrane just pinches in until we form 2 individual cells. Now, we now come on to the idea of cell differentiation. So what we're looking at then is we've started off with these unspecialized cells, and in order to actually make organisms more efficient, we need specialized ones. So we need to convert those unspecialized cells into specialized ones. Therefore, that is differentiation, just converting unspecialized cells into specialized ones. Good examples of this, your sperm cells, where they've become adapted obviously to swim to the egg. So they've got things like lots of mitochondria to produce the energy for movement. You've got an acrosome in the sort of head of them there in order to break down the membrane to allow it to get into the egg. Red blood cells, loads of specializations which we're going to come on to later on. Before we can actually have this process of differentiation, we need to have undifferentiated cells in the first place. And these are our stem cells. So if anyone ever asks you what is a stem cell, it's an undifferentiated cell. We have 2 types of stem cells we need to know about, embryonic and adult. Embryonic stem cells are the ones that are found in embryos, as the name suggests, and they can differentiate into all cell types. Adult stem cells, these are obviously not found in embryos, found in adults, and they can only differentiate into some cell types. So there's a limitation with the adult stem cells in that they can only become some cell types, whereas the embryonic can become all cell types. If we want to know the name of our stem cells in plants, that is a meristem. So if they ask you what are the stem cells in plants called, meristem is all we need to put there. We'll find those meristem cells in the growing bits of the plant, so the tips of the roots, the tips of the shoots, and any of the buds as well. Now, the next little section is all about the transports and exchanges that go on. So again, this is some of that advanced listed content. First thing, surface area to volume ratio. Hopefully, at this point, we can all calculate the surface area and the volume of a cube. If not, go over that, because they're not going to ask you to calculate the surface area and a volume of a weird shaped cell. They're literally just going to get you to do the maths on a cube. If you're looking at the surface area to volume ratio, remember to give it in its simplest terms. Okay, so don't just leave it as 12:3, simplify it down as 3:1. Okay. Now, surface area to volume ratio is important in our living things because it's all to do with working out whether diffusion is adequate or not to meet the needs. And the larger the organism, the lower the surface area to volume ratio becomes. So eventually we hit a point where diffusion is too slow to supply everything we need. So we need to then develop some kind of adaptations. And this is usually done through transport systems and specialist exchange surfaces. Now, what we're looking at first of all then is the transport system, the circulatory system. Hopefully we know the circulatory system is made up of the heart, arteries, veins and capillaries, and it's going to be all put together in what we call a double circulatory system. So double circulatory system just means that the heart is going to have blood flow through it twice on one circuit round the body. So it's going to be pumped twice by the heart on that one circuit. This means that we can have the actual blood at a higher pressure than we'd be able to in a single circulatory system. First type of blood vessel, the arteries. These ones have a thick muscular and elastic wall because they need to withstand the high pressures. Veins have the thinner wall and a larger lumen, the space the blood flows through, because the blood is under a lower pressure. Because of that, they also have those structures called valves which stop the backflow of blood. Final one are the capillaries, and these are the ones where the exchanges take place. So their walls are only one cell thick to make the fusion happen faster. Back to what we said earlier about diffusion, smaller distance, faster rate. In terms of the bits of the blood we need to know for our B2 topic, the first one are the red blood cells. So what we actually find is red blood cells are there to carry oxygen. Now, they have a few adaptations for this. Firstly, they've got a large surface area to volume ratio, and that's all to do with having rapid diffusion. Secondly, they've got this biconcave disc shape, which increases the surface area to volume ratio. They contain that carrier protein of hemoglobin, which binds the oxygen, and they do not have a nucleus because that leaves plenty of room for the hemoglobin. The second part we need to know about is the plasma, which is basically the straw-coloured fluid that all the blood cells float in, and it will also transport things like the digested food, waste products, antibodies, hormones, etc. We now need to have a little look at the heart itself. So there is a picture of the heart. I'm not going to sort of show you all of it in great detail, but hopefully we do remember there are 4 chambers, the 2 atria at the top, the 2 ventricles at the bottom. The left hand side of your heart has a thicker muscle wall than the right hand side of the heart, and that's because the left hand side has to pump the blood all the way around the body, the right hand side only has to go so far as your lungs. They could ask you the names of the individual valves within the heart, 3 to know. Between the right atrium and the right ventricle is the tricuspid, so think of the RI and tri, the RI and right. Between the left atrium and the left ventricle is the bicuspid valve, and then between the ventricles and either of the arteries leaving, those are semilunar valves. Whole purpose of having valves in the heart is just to stop the backflow of blood again. One point to note is when you're actually labelling left or right on the diagrams of heart on your exam paper, remember it's the opposite to the way you look at it. So you've got to kind of imagine you're lying on your exam paper waving up at yourself. That's the way you actually label it. Plant transport systems next then. We've got 2 types of plant transport tissue we need to know, xylem and phloem. So xylem transports the water. These are dead cells with no end walls that have been thickened on their cell walls by the provision of lignin. Now, what we find is that's going to allow us to transport the water and the minerals. Second type is the phloem. So what we actually find with the phloem then, this is how we're going to transport the dissolved sugars and other food molecules in a process called translocation. Now, phloem are made of living cells and their end walls have become these things called sieve plates. So that's just to allow the sugars to be able to pass through nice and easily. We now need to come on to the process of transpiration, which is the loss of water vapour from the aerial parts of a plant. The transpiration stream is actually the description of the movement of the water. So starting in the roots, through the xylem, and then out of the leaves. Any of the water we lose from the leaves, we then have to replace from the roots there. So we've got this constant movement of water, which also allows us to transport those minerals as well. How does water get into the plant? Well, it enters through the roots, through special cells called root hair cells, and it does so in the process of osmosis. From that root hair cell, it then travels cell to cell until it gets to the xylem, and then once in the xylem, off it goes through the plant. It's going to go all the way through the xylem up into the leaves, and when it gets there, it's going to pass out of the leaves through that structure called the stomata, which are little pores on the underside of the leaf. Around the stomata, we've got those cells called guard cells. So if we don't have much water present, they're closed, but if there is plenty of water, the guard cells become turgid and they swell out, opening the stomata there. The reason they do this is that on this inside of the actual guard cell, so the side closest to the stomata, that cell wall is thicker. So as they actually fill with water, it bends it outwards. If obviously we are losing water from the top of the plant, then we're going to have a lower pressure at the top. Where we're taking water in at the bottom, that's a higher pressure, and therefore we've created a pressure gradient. So as a result of that pressure gradient, water is going to move up through the xylem, always from the roots up to the leaves. Never the other way. Xylem only goes up from the roots to the actual leaves themselves. What we're going to find is that there are some plants that obviously don't want to lose vast quantities of water, and so they have a thicker waxy cuticle on the surface. So top surface of the plant just have a much thicker waxy layer, okay, and that just minimises the amount that we lose through transpiration. There is one bit of equipment that we could see that we can use to investigate transpiration, which is our potometer. So quite simply, stick your little chute in one end. We then have a little scale here with a capillary tube, which is a very small tube. Put an air bubble in it, and then you would have your little plant here for a minute, 2 minutes, and then you'd record how far the little air bubble moves. That then allows you to calculate the rate, just distance divided by time. We do have some factors that affect the rate of transpiration. First one, temperature. Very simple pattern. Increasing temperature increases the rate of transpiration. Second one, light intensity. Increase in light intensity increases the rate of transpiration. However, we do level off because when all the stomata are open, increasing light intensity will do nothing more. Air movement, if it's a windy day, things evaporate quickly. Reason for that is that we have a steeper concentration gradient between the leaf and the air, so that means we get faster diffusion. Fourth one is humidity. Now, humidity is just a measure of how much water is in the air, and this is the one that goes the other way. So what we actually find is that if we increase the humidity, we decrease the rate of transpiration. So the higher the humidity, the lower the rate of transpiration, because it's got a much shallower concentration gradient between the inside and the outside, so that means a slower rate of diffusion. Right, that is all of B2. We are now just on to B3. So B3 is going to start off with the nervous system then. So still for absolutely everyone. When we're talking about the nervous system, we need to know a few key terms here. The word stimulus just means a change in the environment. The receptors are the groups of cells that are going to detect that stimulus. And the effectors are the things that bring about a response. They would be muscles or glands. So muscles will contract in order to move something, glands release hormones. If we think about the structure of our nervous system, we have the central nervous system, or CNS, which is made up of just the brain and the spinal cord. We are then going to have 3 types of neuron we need to know about. First one, sensory neurons. These are the ones that go from the receptor cells to the central nervous system. Relay neurons are the ones that connect the sensory to the motor, and then the motor neurons are going from the central nervous system to the effector. So remember that we have those 3 different types of neuron. What we need to know then are 2 different pathways. So the first one is actually understanding how we kind of have control over certain bits of our body using the brain, okay? First thing, we have our stimulus, which is detected by the receptors. The receptors then send an electrical impulse, get used to that electrical impulse phrase, along the sensory neuron to the spinal cord, and then it sends information up to the brain. The brain is then going to send information back down the spinal cord through a motor neuron to the effector, and that brings about the response. Whenever we're talking about information being sent through a neuron, we're always going to use the phrase electrical impulse. Don't say other things, okay, because it won't get you the marks. And some of these questions on the nervous system can be 4 marks potentially, which are all associated with the words electrical impulse. So if you didn't include electrical impulse, you've just lost 4 of those marks straight away. Harsh, but it's happened in the past. Because this is going up to the brain, this is obviously something that we are aware of, and therefore it takes a little bit longer. Okay, so 0.7 seconds is actually a long time for this. But this does allow our brain to actually put together all of the information and come up with what we refer to as a coordinated response. The second option is that we're going to have something that doesn't pass through our brain. So this is something we're not consciously making a decision about for it to actually happen. So we are going to have our reflexes. Now, reflexes are really important because they're involuntary first of all, and they're incredibly quick. So we're talking 0.2 seconds. We've shaved half a second off of the one we have our brain involved in. In terms of the reflex arc, then what we need to know is there is a stimulus that is detected by the receptor cells. The receptor cells send an electrical impulse through the sensory neuron to the spinal cord. The electrical impulse passes through the relay neuron to a motor neuron, and the electrical impulse passes down the motor neuron to the effector, which brings about the response. Look at the context of the question and then just apply the actual bits there. So rather than just saying stimulus, say what it is, bee sting, stuck their hand in a pot of boiling water, whatever it is, there's your stimulus. What's the response? Don't just say there is a response, say what it is. The arm moves away, for example. So just make sure you know that. And I know that some of mine have literally just learnt the little flowchart that I put on the board. Do not just write the flowchart as it is that you've learnt it. Turn it into vague sentences. So you can't literally get a mark just for saying receptor cells to sensory neuron to spinal cord. You've got to fill in those little words of electrical impulse travels through. So not great detail, but please add a bit more detail than just the little flowchart we learnt. Next up, if you're doing combined science, you can just switch your brains off for a minute because this is a biology only topic. It is the eye, which we all love, I'm sure. So here is the eye diagram that we should all know the key bits on. Start with the easy one. The thing sticking off the back is the optic nerve. That's obviously going to take the information in the form of electrical impulses from the eye to your brain. The retina is the lining all the way around here. That is made up of these light sensitive cells. We then have a ciliary muscle, which is all to do with controlling the shape of our lens here. The suspensory ligaments hold the lens. The lens itself is all to do with focusing the light. The pupil, that hole that's the black bit in your eye, that's allowing the light through. Iris is the coloured bit, and that's got circular muscle that's going to change the size of the pupil. And then across the surface of your eye, you've got your cornea, which is a transparent coating, basically starts to refract the light. So, as far as how the eye works, we need to be able to focus on things at different distances. So what you actually find then is if you want to look at something that is up close, then what's going to happen is your ciliary muscle is going to contract and it makes the lens more convex. So it basically makes the lens fatter. That means it's going to focus the light exactly, in theory, on the back of your eye on the retina. If, however, we're looking at something further away in the distance, the ciliary muscle will relax, the lens becomes less convex or thinner, and it allows us to focus on those distant objects. So as far as being able to focus on things near or far, it's all to do with the ciliary muscle, either contracting or relaxing, changing the shape of the lens. We do have some problems of vision we need to know about. So, first one, short-sightedness. This is where you can see things up close absolutely fine, but look at something in the distance and it's just a terrible blur. Some of you might realise you are short-sighted when you sit in the exam hall for the first time tomorrow and realise you can't see how long you've got left on your exam. Now, that's all caused because your lens is becoming too strong or the eyeball is too long. So basically it's focusing the light in the wrong place in your eye. All we do to correct it is we use a concave lens. If you are long-sighted, it's the opposite. You can't see things that are up close, but you can see things in the distance. So to correct that one, we use a convex lens. Third vision problem is being colourblind, and this is usually a genetically inherited condition, and it does affect men more than women. So we can't fix that one. You are just colourblind, and it just means that you have difficulty making out different colours. Depending on what type of colour blindness will determine which colours you struggle to make out there. In terms of those cells on the retina, we have two types, rods and cones. So the rod cells are the ones that allow you to see in low light levels. They don't respond to different colours, but they do let you see when it's quite dim outside. Your cone cells are the colour cells, okay? So they're the ones that are going to respond to the red, the blue and the green light. So they're the ones that allow us to actually get that perception of colour that we all know. Next up is the brain. So again, we are just on the biology folks here. They could be me and give you a picture of the brain like so and ask you what part X is, because there are 5 we need to know. The cerebrum, the bit across the top. The cerebellum, the bit looks kind of like a leaf. Our medulla is the bit that's kind of in line with your nose. It's probably the easiest way to do that. And then we've got the pituitary gland and the hypothalamus. So the pituitary gland is the bit that dangles down a bit more. The hypothalamus is pretty much dead centre. They're the bits you need to know, okay? Now, in terms of what they do then, the cerebrum is all to do with your complex behaviour. So learning, what we're doing now, memory, your conscious thought, all of that is cerebrum based. Cerebellum is to do with your balance, your posture. The medulla is your automatic actions, so heart rate and breathing rate. Hypothalamus is basically the regulation zone. So this is controlling your body temperature, your water balance. And then your pituitary gland, that's going to store and release hormones, which are involved in a range of other things. For those of you doing higher tier biology, we need to know about investigating the brain. Now, we used to do this by basically just studying stroke patients. Because someone has a stroke, it's affected part of the brain. If you have a little look to see what is impaired in their actual function, and obviously which bit of the brain's affected, it gives us a bit of an idea. We also have used electrodes, which are fun. So we can literally stick electrodes into brains and then just stimulate them by sending small electrical impulses in, see what happens. You can see why people may have a problem with that. These days we have better technology. We can use things like CT scans, which use X-rays to produce 3-dimensional images inside the body. And obviously we can identify any anomalous or odd regions and what behaviour that links to. Obviously good, because we're not sticking things in your brain and sending electrical impulses in. We can use MRI scans, similar way, another type of scan that we don't have to slice and dice. And we can use the rather flashy fMRI or functional MRI scans. Now, these ones literally show the bits of the brain with increased blood flow as someone does an activity. So they'll literally get someone to sit there and play a guitar, for example, and monitor which bits of the brain are flashing up with more blood flow to them. That's how we know which bits of the brain are associated with certain activities. Now, there are difficulties with studying the brain. First of all, we need consent from the patients to share their information, and people might not want to do that. So they may sort of think that actually, I don't want everyone knowing all the inner workings of whatever accident I had, for example. To get a reliable conclusion, we need many case studies, and we don't always have a large number of that same situation. And obviously, if we're thinking animal testing, we've got the ethics side of things as well there. Still just on the higher tier, folks, then we need to understand that when we talk about the nervous system, we've got the central nervous system, brain and spinal cord, peripheral nervous system, all of the neurons that connect to the rest of the body. Depending on what bit's damaged is going to determine the severity and what we can do with it. So if we have damage to our central or peripheral nervous system, this could have been caused through an injury, could be a genetic condition which just leads to obviously breaking down over time, could be a disease, or it could be some kind of toxin that we've consumed. Now, if we affect the peripheral nervous system or damage that in some way, that's usually affecting either the sensory or the motor neurons. So this might prevent us from detecting pain. It might lead to areas that are numb. It might lead to loss of coordination. Quite often, these might be able to heal if they're very limited in the damage. Severe damage obviously is not going to be able to regenerate, but limited damage might be able to regenerate. If we have more severe nerve damage, then there is an option for surgery in some cases where we're just going to graft tissue over the damaged section, but it won't always work. If we're talking about more severe damage, then we could lose control of entire body systems. We might be talking paralysis, either partial or complete, and there could obviously be quite serious neurological issues as well, memory loss, processing difficulties, etc. And the key thing to remember is if we're damaging our central nervous system, then that does not regenerate. So that is permanent unless we can use some form of surgery to correct it, and those are limited. So why is it so tricky to repair things like spinal cord injuries? Well, first of all, your spine is a grand total of 1.5 centimetres in diameter. So your entire spinal cord, just 1.5 centimetres. And in that 1.5 centimetres, we've got 31 pairs of nerves. So finding the individual damaged fibre and repairing it without damaging the others in that tiny little space is actually incredibly hard. Hence why if you've done damage to your spinal cord, it's usually irreversible. In terms of what we can do for damages to the brain, then if it's a tumour related one, chemotherapy, radiotherapy, we could use surgery to remove damaged tissue in some places, not others. If they're right in the centre of the brain, you damage healthy tissue, or we could use electrodes to stimulate brain function in some areas. Okay, we're now back to absolutely everyone again as we come on to hormones then. So this is foundation, higher, combined, triple, you name it, everyone's involved. Hopefully we know a hormone is a chemical messenger and they are made in the endocrine glands. Any hormone that we make is going to travel through the bloodstream. So if they ask you how does the hormone get from wherever to wherever, through the bloodstream is the answer. And they're going to bring about a response in the target organs. They could ask us to compare the hormonal versus the nervous system. So hormones are slow, nervous system is fast. Hormones are longer lasting, nerves short lasting. And then what we also find is that the hormones tend to target a much larger area, whereas the nervous system is precise. So we do have those comparisons we can draw. Do remember when we're talking about target cells, these are the ones that have receptors for that particular hormone on their membrane. So the hormone binds to the receptor and brings about a response in that target organ. If you're doing the higher tier, then we need to know about the process of negative feedback. This is just a control mechanism that we use in the body in order to actually maintain this process of homeostasis. So homeostasis is the maintenance of a constant internal environment, and negative feedback is how we do it. So we've got receptors that detect these small changes, and then we're going to reverse that change using effectors. So we keep everything within this very narrow range of our normal level. You need to know of two kind of key examples, if you like. First one is thyroxine. So it's produced in the thyroid gland, and it regulates that metabolic rate of the body. So it's all to do with the energy production in your body, basically. How it's made is your thyroid gland is going to take iodine, which you get in your food, and it converts it into thyroxine by joining it with an amino acid. Now, what we need to understand then is this negative feedback loop that we get here. First thing, we're bringing in our knowledge on the brain then. So the hypothalamus, the region that detects this, is going to detect that there's a low level of thyroxine in the blood first of all. That then sends a bit of information to the pituitary gland saying release TSH. That's the hormone TSH. TSH travels through the bloodstream, and it's then going to stimulate the thyroid gland to make thyroxine. So the thyroxine levels then go up. Hypothalamus detects that we're at a normal level of thyroxine, sends the message to stop the pituitary gland making TSH, so the thyroid stops making thyroxine, and as a result, we then start to drop again. So we're constantly just fluctuating between that slightly high, slightly low, but always within the accepted range. So thyroxine made when obviously we release our TSH from the pituitary gland to stimulate the thyroid. The second hormone we should be aware of is adrenaline, produced in the adrenal glands, and this is all to do with those fight or flight responses to stress. So if we've got adrenaline, then this is going to lead to faster respiration, increased heart rate, increased breathing rate, and it actually diverts blood away from areas like your digestive system. So what we find here is, and there was a question, I think it was about 3 years ago now, about why we'd apply adrenaline onto a cut. So the whole idea there is it's going to help to divert the blood away, so it stops you basically bleeding to death. Right, another favourite, this is for everyone, okay, so combined, triple, higher foundation, you name it, we are on the menstrual cycle. So, hopefully we do know the menstrual cycle is a monthly cycle getting the body ready for pregnancy that lasts around 28 days. Here is our lovely little diagram of the female reproductive system. So we have 2 ovaries, they're connected by the fallopian tubes onto the uterus here, and then you have the vagina at the bottom, obviously separated from the uterus by the cervix. Now, in terms of what's going to happen, in a typical month, then obviously the lining of the uterus starts to thicken just in case a fertilized egg was going to be in existence. So as that uterus lining starts to thicken, an egg is going to be maturing in one of the ovaries. 14 days into our cycle, so day 14, is the point of ovulation. So the egg is released from the ovary and starts to travel along the fallopian tube. If sperm is present at that right time, then it may fertilise the egg in the fallopian tube. That fertilised egg continues on down and then may implant into the uterus lining. That's where it will stay, grow, develop, form a little baby, and happy times for all, hopefully. Obviously, that doesn't happen every single month. Just as well, there'd be way too many people. Instead, if that egg is not fertilised, then all that happens is the lining of the uterus and the unfertilised egg are removed from the body in the form of a period. So, we have 4 hormones we need to know. So we're going to go through these 4 hormones in turn, see what they do, where they're produced, etc. So our 4 hormones, FSH, estrogen, LH, and progesterone. Now, first one, FSH. You don't need to know the full name, follicle stimulating hormone. The abbreviations are perfect, okay? So FSH is secreted by the pituitary gland, and it's going to travel through the bloodstream to the ovaries where it causes the egg to mature. Now, a second part of this is it's also going to stimulate those ovaries to produce estrogen. So FSH made in the pituitary affects the ovaries, egg matures, and estrogen is going to be produced. Secondly, estrogen then. This is going to be made in the ovaries, and it's going to cause the uterus lining to thicken. Now, what we actually find here is that the increasing levels of estrogen actually inhibit further FSH production. And that's good, because it means that what we're going to do here is actually stop more than one egg from maturing. Usually. Not perfect, but not bad. So what that means is you won't have several babies all at once, in theory. It also stimulates the pituitary gland to release LH. So estrogen made in the ovaries causes the uterus lining to thicken, and it's going to inhibit FSH, but it also then releases LH through stimulating the pituitary. Luteinizing hormone is next, or LH, produced in the pituitary gland, and this one, when it peaks in the middle of the cycle on day 14, is when ovulation occurs. So LH causes ovulation itself. Last hormone is progesterone. Now, progesterone is the one that is made by the corpus luteum. Lovely bit of Latin they threw in a few years ago. You can describe it as an empty follicle if you prefer. Okay, so if you can't remember the wonderful Latin corpus luteum, just go with empty follicle. Works just as well. Now, the whole purpose of progesterone is just to maintain that thick uterus lining, and those levels have to remain high throughout all of pregnancy. Progesterone also inhibits further LH production, so it prevents any further ovulation from occurring. Okay? If they're being mean, they're going to chuck in this little graph in some form. It looks horrendous, but it's not as bad as it seems, because actually all we've got are 4 individual graphs. Just break them down one at a time and take what you know about the hormones to then basically use the graphs to help you explain it. So, for example, we've just said that where LH peaks on day 14, that is ovulation. So here we go. If we have a look, there's our little peak for LH, the blue line. If you track all the way down, you can see day 14, egg released. Okay, they obviously wouldn't have egg released there, but day 14 does tell you what's happening. So you can look to see what's happening with the lining, look to see what's happening. So obviously here we see progesterone going up, the lining stays thick. We've got that correlation. So you can literally kind of take what you know and not necessarily read too much on those graphs. So don't be thrown by them. Nice, hopefully simple bit next is all about controlling reproduction. So basically it's the contraception bit. Hopefully something we are all well versed in, being roughly 16-year-olds. So contraception is just the technique we use to avoid pregnancy. 2 types, hormonal and non-hormonal. Hormonal are going to use hormones to disrupt the cycles, whereas non-hormonal are barriers. They basically stop the sperm reaching the egg. So know a couple of examples. Condoms, for example, are always a good go-to for the non-hormonal ones. And don't mix up the IUD and the IUS. So IUD, non-hormonal, IUS, hormonal. So just go careful on those ones not to mix them up. Most likely question they'll give you on this is a table where they're going to have the effectiveness of different treatments and ask you to evaluate them. So on those, all you got to do, pick out the one that is the highest percentage, because that's the most effective, and then obviously sort of talk about why it might be best. Do consider things like, if you've got to take a pill every single day, that may not be great for someone who's perhaps disorganized. So obviously going down the route of a condom might be better in that case. For the higher tier folks, we then need to know how we can actually use hormones to treat infertility then. So as far as infertility goes, then we have a range of different causes for it. Could be blocked sperm ducts or not enough sperm being produced if we're talking about issues that men could have. It could be that the ovaries aren't producing enough mature eggs, or they're not releasing them as they should. So we have a range of kind of issues that could be a cause of infertility. What we generally do is we use FSH as one of those key fertility drugs. So if they ask you to suggest the name of a fertility drug, FSH is great because it stimulates eggs to mature. Now, because of that, we're going to be able to create a whole lot of eggs at the same time, basically. When we're then ready to collect, you get a dose of LH, the one that causes ovulation. So that's your trigger shot. So what happens is when you first go through this process, you'll be taking FSH to cause a large number of eggs to mature at the same time. You then have the LH causes those eggs to be released, and they're basically collected. At that point, they go into a little petri dish, mixed with the sperm in the little petri dish, and then they watch them. So they'll have a little look as they hopefully fertilise, and they start to divide. Once they've formed this early embryo, they usually let it go for about kind of 8 days roughly. Then they will implant the kind of pick of the litter, if you like, into the uterus of the mother. Normally only 1 or 2, and then hopefully that leads to pregnancy. Not a guarantee, but hopefully. There are some issues with IVF though. Some people argue it's not natural, therefore we shouldn't do it, of course. But there are some positives here. It does mean that parents who would not otherwise be able to have a baby can have one. It does obviously give people a chance to have a baby later in life if obviously they weren't ready when they were younger. Downsides, I suppose, is that it's very expensive. So you can get some treatments on the NHS, depending on where you live, determines how many you get. And then if you have to go private, be prepared to pay over £10,000 for this per round. Okay, so it's not cheap at all. And you can end up with multiple births as well. So depending on your viewpoint, a positive, a negative. Obviously, if we're going for the negatives, you may not be ready for having maybe 2 babies at once. If you're going with the positives, buy one, get one free. Wonderful. On to our plant hormones now then. So, plant hormones, we need to understand the tropisms. This comes up in our higher tier for the experiment section. So, tropism, quite simply, is growth in response to an external stimulus. So, plants can respond to their environment, okay? We have phototropism, all to do with light, gravitropism, to do with gravity. Now, this is auxins, are the hormones that control this. So auxin is going to be made at the tips of the roots, the tips of the shoots, and what happens is it's going to affect cell elongation. So, if we think phototropism first of all, this is the plant's response to light. If we're talking about the shoots, they demonstrate positive phototropism. They grow towards the light. If we're talking about the roots, negative phototropism, they grow away from the light. And the way that works is that in terms of our actual plants, the auxin is produced in the tip, it diffuses down, and it accumulates on the shaded side. As a result of that, the cells then elongate, so they get longer, and if this is our shaded side, what happens is it bends it towards the light there. For gravitropism, what happens is positive gravitropism, same direction as gravity, negative gravitropism, away from gravity. So this is all to do with making sure that our shoots grow up and our roots grow down. So what we find, the auxin is going to accumulate on the side closest to the ground due to gravity. And as a result of that, it's going to obviously stimulate cell elongation in the shoot. So it's going to grow upwards there. And it's going to inhibit cell elongation in the root. So it bends that one downwards. If we wanted to investigate gravitropism, we use a bit of equipment called a clinostat, which basically is a little plastic pot that you stick your seed into that spins, because that then eliminates gravity from the actual thing. Now, we need to know uses of hormones. Hopefully we know auxins are all to do with stimulating growth by causing cell elongation, and we can use them to help regulate the development of fruit. Ethene is going to cause fruit to ripen, and gibberellins are going to end the dormancy of seeds. So some specific examples for the higher tier folks. You need to know that selective herbicides, i.e. weed killers, these contain auxins, and they're going to kill broadleaf plants, not narrowleaf plants. We can use rooting powders, they contain auxins, and that basically means our cuttings will grow roots. We can spray auxin on fruit trees to delay ripening, so that means we can collect the whole harvest at the same time. We can then use ethene to spray on fruit trees to make fruit ripen quicker, so that means we get fruit earlier in a growing season. We can carry out this process called parthenocarpy, i.e. making seedless fruit. Again, take auxin, stick it on an unpollinated flower, and it produces seedless fruit at that point. And we can obviously break dormancy. So we can spray seeds with gibberellins to trigger germination to allow us to grow them all year round. Now, one thing that's quite obvious from all of that is if in doubt about which plant hormone does something and you really can't remember, just write auxin, because it's right in virtually all cases. So if in doubt, go with auxin. The last section is all the controls stuff. So first one, controlling body temperature. Hopefully we know that human body temperature is 37 degrees normally, and we need to keep it at that temperature because of the enzymes. If we get too cold, your core body temperature is going to drop, enzyme reactions are going to slow right down, we won't get enough energy through respiration, cells will die. If we get too hot, so over 40 to 42 degrees, enzymes may denature and therefore you may die. There's a recurring theme. In order to control this, we have receptor cells that monitor both the external temperature and the temperature of your blood. That information is sent to the thermoregulatory centre in your brain, and then we're going to have various mechanisms brought into play to make sure you stay within that narrow range around 37. So if you are too hot, what's going to happen is any of those hairs on your arms, for example, are going to lie flat. That prevents air being trapped, and that means you are going to transfer more of your heat from the body to the surroundings. You might sweat, and then as the water evaporates, we're transferring energy from your body to the surroundings, and we experience vasodilation. So the blood vessels are going to widen near the skin. This means we've got an increased blood flow through capillaries, and that means more heat is lost by radiation. If you're too cold, it's the opposite. Body hairs rise, trap air against the skin, insulates the body. You don't sweat, vasoconstriction occurs, so that means we reduce blood flow and heat loss, and you might shiver, which basically means cells respire faster, and therefore we transfer extra energy to make you warmer. Next one we need to understand is controlling our blood sugar levels. So hopefully we know this is going to come down to one hormone. If you only know one, the one is insulin. So what we have is inside our pancreas, we are going to detect if the blood sugar levels are going too high, and as a result, insulin is released. Travels through the bloodstream to the liver and the muscles, and it's going to convert glucose into glycogen for storage. If you're doing the higher tier, you also need to know about a second hormone here, glucagon, and the glucagon is what's going to convert the glycogen into glucose. So what happens is if your blood glucose level is too low, then the pancreas releases the glucagon, which then converts glycogen into glucose to bring it back to normal. So foundation, you only need to know insulin, higher, insulin, and glucagon. Please don't mix up all the glue words in here. So glucagon, glycogen, glucose, make sure you've got them all straight. We do have two types of diabetes where we don't manage to control our blood sugar levels very well. Type 1, where we can't produce insulin, usually because we've destroyed the cells that make it in our pancreas. So what we actually find there is that's usually controlled by having to have insulin injections. But you also need to make sure you've got a balanced diet and you exercise. Type 2 diabetes, our body's not responding as it should do to insulin. It might be it's not making enough, or it's just no longer able to respond as it used to. Can be controlled by balanced diet and exercise, but may need injections of insulin as well to help. The last one is controlling our water balance, which brings us on to the kidney. So what we actually find then is if you've got too much water in your blood plasma, we undergo lysis with our blood cells, which would be bad. Too little water, then they're going to start to shrivel up, and that also would be bad. So we need to keep our water levels just about right. So the way we do this is by using the kidneys. Now, what we actually have then are three key sections in our kidney. You've got the capsule around the outside, the cortex is this outer layer, and then the medulla is this kind of inner region here. They're the only three bits you need to know for the GCSE. Inside the kidney, we've got about 1 million of these individual filtering units called nephrons. Now, the nephron itself is made of several little parts, which we're going to go through now. Firstly, the blood is going to enter the nephron through this little bit called the glomerulus, which is basically a knot of capillaries. Now that glomerulus sits inside a structure called the Bowman's capsule. So as the blood enters the glomerulus under a high pressure, it forces out these small things, so things like our individual ions, etc., the urea, out of the blood and into the Bowman's capsule. Okay, anything that's too big, blood cells for example, remain in the blood themselves. What we're seeing there is a process called ultrafiltration. So quite simply, what happens is that high pressure in the glomerulus forces out small molecules, but not the large ones. Hence ultrafiltration, we're filtering it. That then passes into our PCT, proximal convoluted tubule, and as it passes through that region, it undergoes a process called selective reabsorption. Now, selective reabsorption, quite simply, is where we're going to reabsorb key bits, okay, the bits we want. Things like the glucose, for example. After that, it continues on down through this region called the loop of Henle, and the loop of Henle is closely associated with the collecting ducts. And it's in that region that we see the extra salt and water being reabsorbed. What we find then for higher tier folks is you need to know the negative feedback control of this using ADH. So we're going to have the hypothalamus detecting the water potential of the blood. If the water concentration is too low, that sends a message to the pituitary gland, which releases the ADH. That ADH travels through the bloodstream to the kidney, and it then basically gets the tubules to reabsorb more water. It makes them more permeable, so more water is going to be taken back into our actual bloodstream. That restores the water to normal, and obviously the hypothalamus detects the water potential of the blood is back to normal, sends the message to the pituitary gland, stops releasing the ADH. The opposite is true if we had too much water. Pituitary gland is then going to say less ADH is released, less ADH is travelling in our blood, kidney tubules reabsorb less water, so more of it passes down to the bladder as urine. We do need to obviously bear in mind that if you don't actually take on enough water, then you are at risk of dehydration. And dehydration is bad because it can kill you, okay? Early symptoms, lack of energy, headache, dizziness, prolonged dehydration, damage to things like your kidneys, your liver, and death. So drink water semi-regularly. Now, the last little bit is 3 terms we should know, all to do with solutions associated with osmosis. So hypertonic, hypotonic, and isotonic. If something is hypertonic, that contains high levels of glucose and salts. So this would be something that you would drink before a race, for example, because it's got lots of glucose in there to get you ready to go. Hypotonic, low levels of glucose and salts. And isotonic should be the same concentrations of ions as in your blood plasma. So you drink that one after a race because it's not going to affect your osmotic balance. And that is all of B1-3. 3 minutes over, but not too bad. So, obviously that's everything in a nutshell. If there are any bits that you are still not 100% certain on, I would have a little look over them tonight before obviously you go to bed. But do remember, don't stay up all night revising because that's actually counterproductive, because if you're absolutely exhausted tomorrow, your brain's not going to function too well. So make sure that you do get some proper sleep. Remember to take a scientific calculator with you tomorrow. Please draw graphs in pencil because you only get 1 bit of graph paper. If you mess it up, that's it, you've blown it if it's in pen. And obviously remember, when you're writing any of your sort of first letters, so the ABCDs, please be careful with your B's and D's. Make sure they really are clear which letter you're going with, because if a marker is in doubt about it, you just don't get the mark. So really be explicit, okay? So that's it for tonight. We will continue with our daily boosters over the next few days, and then night before your chemistry paper, we will be back again. So good luck, everyone. I hope you do well tomorrow, and we shall see what the paper brings tomorrow. Have a good evening.